So starting with anatomy, I think it's a good idea with the esophagus especially to start with some histology. The reason why I think that's a good idea is that, as we talked about in the GI intro lecture, most of the GI tract is formed from the embryonic GI tract, and with that comes its own cell types. With that said, the embryonic GI tract doesn't begin until the very end of the esophagus. So for most of the esophagus, it's actually composed of a different cell type. It's has a different arterial blood supply, and that can present with different clinical conditions like different cancer types. And right here is another reminder from our GI intro lecture that not only the upper end of the esophagus, but also our oral cavity and the very end of our anal canal, the lower half of it, is all technically not part of the embryonic GI tract. So it has different cell types composed in that region. And that cell type is referred to as non-keratinized stratified squamous epithelia. And so I'm going to break this phrase down so we know exactly what that means. So we saw epithelia in our oral cavity lecture. Epithelial cells are just cells that line glands. They could line organs. They can line our skin. So it's a pretty broad term, and it's densely packed, way more densely packed than connective tissue. And you can see there's different types of epithelia, and I'll show you a picture. We have two different types of epithelia, and in both examples, you can see how tightly packed these cells are compared to the connective tissue that exists right behind it. Now, when we refer to whether something's keratinized or non-keratinized, that's usually to refer to squamous epithelium, and I'll show you what I mean. So keratin is a waterproof protective protein that exists on our hair, our skin, and our nails. And if you're non-keratinized, that means you don't produce keratin. So why do they have this distinction in this phrase when it comes to esophageal epithelium? The reason they do that is because there's a different type of stratified squamous epithelium that exists in our skin that is keratinized. And let me show you a picture. So in our skin, we have this thick layer of keratin here. And again, that's a waterproof layer. But in the esophagus, and then you can see on the left side here, you can still see the stratified squamous cells below this thick layer of keratin. Whereas on the right, if we look at a picture of our esophagus, we still have this layer of squamous cells, but we don't have that layer of keratin. That's why it's called non-keratinized. And the last thing, which I kind of alluded to a moment ago, is stratified. So cell types can be simple or stratified. Simple just means there's one layer, and stratified means there's many layers. And so if we go back here, you could see how many layers of squamous cells are just packed on top of each other, going all the way through. You can see probably 24 layers of cells right here. And then the last thing I just want to clarify is what it actually means to be a squamous cell. What does that look like under a microscope? Well, we've been looking at some of these cells, and they have this fish scale appearance, which is pretty classic. On the left, we have columnar cells. They don't really look like fish cells. They look like columns, so they're aptly named. But these guys over here on the right are squamous cells. They look like fish scales, excuse me. And you can see that in our upper part of our esophagus, you're going to have more of these types of cells. And in our lower part of our esophagus, you're going to have more columnar epithelia. And so, and the relevance of this, like I just alluded to, is that when we look at our embryonic GI tract that starts at the very end of our esophagus, you're going to see columnar epithelia. But there are circumstances, which we'll talk about later, where our squamous epithelia can actually convert into a columnar epithelium due to repeated damage to the tissue. And that may not be dangerous, but that columnar cells that have already changed from a different cell type, those could morph even more into a cancer. So that's always something we have to watch out for. Now let's talk about a few relevant features of the esophagus. It's just a hollow tube, so there's not too much to talk about. When we discuss motor control, I just wanted you to know that the upper 1/3 of your esophagus is composed of striated muscle. And what that means is that you can voluntarily control it, kind of like when you're taking a bite, you can feel the upper part of your throat being able to control that swallow reflex. But then as the food passes down the esophagus, you lose control, and that's because the lower 2/3 of your esophagus is composed of smooth muscle. I think this is a good part to talk about the diaphragm, since the esophagus does pierce the diaphragm, and they do like to test which structures pierce the diaphragm. So let's just go over that really quickly. The esophagus pierces the diaphragm at the level of T10. And so if this is our diaphragm here, and then I just wanted to refer to some other structures that pierce the diaphragm too, including their spinal level, and I'll just give you a mnemonic that I've always remembered. I don't know how useful it might be to you, but whenever it comes to anatomical structures and stuff, I do find it a little easier to remember it through mnemonics, since there's often not a clinical correlate to help you out. So the IVC pierces the diaphragm at the level of T8. The esophagus and the vagus nerve pierce the diaphragm at the level of T10. And then three structures pierce the diaphragm at the level of T12. That's the esophagus vein, aorta, and thoracic duct. And so I've always used this mnemonic, that I made up. It just goes I8, 10 EVs at 12. So EV is a Pokemon. It's kind of a sad mnemonic now that I think about it, but it is what it is. And so I think of the 8 for the IVC is the 8 for I8. And then 10 EVs is T10, which refers to the esophagus and the vagus nerve. And then for T12, I think of the azygous vein, aorta, and thoracic duct. I don't know if that'll help you, but that's just kind of how I remember the structures that pierce the diaphragm. And the only other relevant anatomical structure that I wanted to go over is the lower esophageal sphincter. Now, this is the sphincter that subdivides our esophagus and our stomach, and it can protect against the reflux of gastric acid into the esophagus. So it plays a really important role. And if we look at here, if you think of gastric acid coming on through to the esophagus, fortunately, we usually have a lower esophageal sphincter that can protect us from that process. Now let's briefly talk about vasculature for the esophagus. The esophagus, you can divide their arterial supply into thirds. So we have an upper third, a middle third, and a lower third. The upper third receives branches off of the inferior thyroid artery. The middle third receives branches directly off of the aorta. And the important one to know is that the lower third of the esophagus can receive branches from the left gastric artery. And why this is important is because the left gastric artery arises from the celiac trunk, which is considered part of our foregut. Anything in anything with the blood supply from the celiac trunk, SMA, or IMA are considered part of our embryonic GI tract. And so this is why the lower esophagus is considered part of the foregut. And I can show you that right here. We have our celiac trunk located right here. And you can see there's 3 different branches right here. And if I show you the left gastric, so this left gastric can come off, and esophageal branches of that left gastric can provide some blood supply to the very last half or the lower third of the esophagus. And there's one clinical correlate that's good to know. I'm going to hammer this home in a few different lectures, including the liver lecture, is that the esophagus drains into both the systemic and the portal circulation because it's part of the GI tract and it's also not part of the GI tract. So for the systemic circulation, you get lower esophageal veins that can drain into the azygous vein, and that can drain into our IVC, which is kind of our main vein that funnels fluids back to the heart. But at the same time, we have some of our esophageal capillaries drain into left gastric veins, which then drain into the portal system. So that goes portal vein, liver, hepatic vein, and then finally to the IVC. And why this is important? This is important because these 2 types of veins, the esophageal and the left gastric veins, can connect at times. Okay, and if there is a backup or a buildup of pressure in one of these veins, it can actually push into the other. So I'll show you what that looks like. Basically, here's what we just talked about. We had our lower esophageal veins that were going into the azygous vein and ultimately into the IVC. Whereas here we had our left gastric vein that can go into the portal vein, and then it goes to the liver, hepatic vein, and ultimately ends up in the same place. Well, these 2, if this gets bigger, if our portal vein gets really big, because of the connection between the portal system and our regular circulation, this vein might get enlarged as well. And if you look here, this is taken from the liver lecture, talking about a few different areas in our body where our portal system and our systemic system have connections. You can see here we have esophageal varices, is what I'm about to talk about, with our left gastric and our esophageal veins. So what does that look like? What that looks like is if we do have a buildup of fluid in our portal system, let's say our liver is not working as well as we'd like it to, sometimes fluid can back up, it'll back up in our portal system. And because of this connection between our portal vein and our esophageal vein right here, it could actually back up and start pushing into this esophageal vein. The problem with that is that these veins here were not built to handle these kinds of pressures, right? So you can actually get upper GI bleeding, and it could be life-threatening upper GI bleeding in some circumstances because of this connection. And you can see a esophageal varix on the right here. You could actually see a few of them, but I'll point out one right here. So that's always something to keep in mind. Now we'll talk a little bit about physiology. And the big thing that you need to know physiologically is how the lower esophageal sphincter works, because there's going to be a few conditions that come up later on when we talk about conditions like achalasia, especially, that are dependent on the physiology of the lower esophageal sphincter. So as we talked about before, this is located between the lower esophagus, excuse me, and the cardia of the stomach. Whoops, give me 1 second. There we go. It protects us against the reflux of gastric acid into the esophagus. And this relaxation is mediated by nitric oxide and a VIP. Okay, so two different compounds that help mediate the relaxation or opening of the sphincter. And both of these, the myenteric plexus can regulate the tone by using these. So I'll show you what that looks like. We have our stomach here, and we have our lower esophageal sphincter. Myenteric plexus neurons can release some of these compounds, and these compounds can act on the lower esophageal sphincter, and it can relax it and open it up. And we're going to find out later that you can have abnormal tone in things like achalasia, even things like GERD. It's actually for the opposite reason, but you can have a few different problems with your lower esophageal sphincter that can cause problems. Thanks for watching. I hope you enjoyed the video. Please like, comment, and subscribe for more content.